Okay, let's see. Okay, so you said this class ends at 4:50? Okay. We'll get as much of this done as possible. By the way, one thing I want to say is for now, I haven't set official office hours. I will have office hours. So not only will you be able to contact me through the Past Tutoring Center, through YouTube, through email, whatever, I'm going to have office hours. Okay, I haven't decided when they're going to be. I haven't gotten definitive proof of when what my schedule is. But for now, what I'm going to do is if you have questions after lab for at least today, I will have 3 office hours literally after this class from 5 to 8. Okay? So for this week, that will be my office hours. So I'm going to get as much of this done as possible. You're welcome to leave at 4:50 when this class ends. But for those of you who really want to hear this, that you'll hear this in lecture, but for those of you who want to hear it, because my office hours will extend past class today, I will essentially go through this, okay, if you want. Okay, but the basic idea here is that we are going to talk about transcription. Okay, you plug this in. Plugging in. Okay. So that's the idea. We're talking about transcription. Okay. So, unfortunately, I can't write on this. I'll get that fixed, but I'll try to talk you through this. Okay. So, transcription is a process by which the cell uses the DNA in the nucleus to make RNA. Okay, so here's the central idea. Let me move this over. Here's the central idea behind gene expression that I was mentioning earlier before I got on a tangent. Okay? If you've taken any kind of biology, you've likely elucidated this to some extent. If you've taken microbiology, I tutor that. I know they go over this to some extent. Okay? So here's a cell. Okay? What we're going to do now is we're essentially going to neglect all the organelles. We're only going to focus on one organelle. That's it. Okay? So you have to imagine there's a cell here with all these organelles. All I'm concerned with is this guy, which is the nucleus. That's the only organelle I'm concerned with. Okay? Does anyone remember what the important polymer is that's in the nucleus? Anyone remember from micro or biology? What's the abbreviation, the acronym for deoxyribonucleic acid? DNA. Right, so what's in the nucleus is essentially lots and lots of DNA. Okay? So DNA, I'm not going to go in a whole lot of detail about it. We'll see a sample strand of this in a few minutes. Okay? But suffice to say, DNA is a polymer of monomers known as nucleotides. So I had originally from our earlier discussion of proteins. Proteins were polymers, right? And their monomers were amino acids. Does that make sense? Likewise, DNA is also a polymer, but it's a polymer of different monomers, and those monomers are known as nucleotides. Okay? So you string a whole bunch of nucleotides together, and you get genetic information. Okay? So here is sort of the central idea behind DNA. So what I'm going to do right here up on the board is let's pretend that that's a DNA molecule. Okay? Well, if I get different colors, let's say that's a DNA molecule. Let's say that there's this component right here, this segment. Let's call this a gene. We'll call it gene 1. Okay? Then, let's say I go over a few nucleotides, and I get to this segment right here. I'll call that gene 2. Okay? And if I had more colors that worked, I'd go beyond that. Okay? What a gene essentially is it's a segment of nucleotides in DNA that codes for a protein. Okay? The next couple lectures are going to be talking about how we do that. So here's the idea from a very abstract perspective. Gene 1, through a series of processes, will code for protein 1. Does that make sense? Likewise, you can make the same argument for gene 2. Gene 2, through a series of processes, will code for what? Protein 2. And, let me do another example. If you go over a few nucleotides, I have gene 3, what does that code for? Protein 3. Okay? So the central idea behind gene expression is you have genetic information in the form of DNA in the nucleus, and through a series of processes called gene expression, you get out proteins. Okay? Now, one thing you should remember from any kind of biology class is that when we're talking about mammalian cells, they have a nucleus. Okay? So we are mammals, we are eukaryotes, so we have a nucleus. And inside the nucleus, this is where I find the DNA. So all genetic information, for the most part, the mitochondria does have some DNA, but in general, the nucleus is where the DNA is. Okay? Does that make sense? Now, this poses a problem. And the problem is as follows. There's an enzyme out in this region. This region, by the way, is called the cytosol. Okay? Basically, any fluid that's not in the nucleus is the cytosol. The fluid outside the nucleus, but within the walls of the cell, that's called the cytosol. Does that make sense? Make sense? Okay. Now here's the problem. DNA is confined to the nucleus. It's stuck there. Okay? DNA is stuck. It can't leave. There's no way that this DNA can exit the nucleus. But if you want the gene to code for a protein, the enzyme called a ribosome is in the cytosol. So the problem is that somehow I've got to get the message from the nucleus out of the nucleus and into the cytosol where the ribosome is. Do you see how that's a problem? Because I can't just simply take the DNA out of the nucleus and make it code for a protein when the ribosome is out in the cytosol. That's a big problem. Okay? So there's an ingenious way that cells get around this. Okay? The DNA in the nucleus, there are enzymes in there that cause the synthesis of a transient carrier of the genetic information known as RNA. Okay? So what this slide elucidates is that there's multiple kinds of RNA. Okay, so notice I mentioned transcription, which is this process that I'm about to describe, makes mRNA, which is also called messenger RNA. There is rRNA, which is called ribosomal RNA. There's tRNA, which is transfer RNA. And there's many more that we're never going to concern ourselves with in this class. The point I want to make is that transcription is a process by which we take the DNA and make RNA from it. Does that make sense? Okay. So, what I'm going to do through a series of processes, which we'll elucidate more later in detail, is I'm going to take this DNA in the nucleus and I'm going to make something referred to as mRNA, messenger RNA. Okay? The process by which I make mRNA from DNA, this process, this is called transcription. This is transcription. In fact, the process by which I make any RNA from DNA is transcription. Okay? So, now I have a different story. Now I don't have this problem anymore because initially I couldn't take the DNA out of the nucleus to the ribosome out here. So what I do instead is I make this carrier of genetic information and I transport that out of the nucleus and to the ribosome. And then the ribosome can use this mRNA to make the protein. Does that make sense to everybody? Okay? So the way that mammalian cells get around this problem of DNA being in the nucleus is they synthesize an mRNA, which, by the way, as we'll find, is an exact copy of the DNA, except for a few things. And the mRNA gets transported out, and the ribosome translates it into a protein. This process right here, in fact, let me do this. I'll call transcription process 1. This process over here, by which the ribosome uses the mRNA, this is called translation. This is the topic of week 2. We're not going to talk about translation today, but understand that the process by which a ribosome makes a protein using mRNA, this is called translation. Okay? That's week 2. So here we're just really concerned about making the mRNA. Okay? So the process by which we make the mRNA from DNA is called transcription. So let me go to the next slide. Oops, I unplugged something. What happened? Something happened. Something come unplugged. Well, I don't know what happened. Oh, screensaver. Okay. Okay. So moving on. Okay. Now, this is a big concept for you to understand. So, what I'm going to do is I'm going to show you a sample of a DNA strand. Now, you can certainly go watch videos on this. I have some. I'm going to have more. I'm going to point you in the direction of some that are really important before I get them up. However, DNA is double-stranded. Okay? Does everyone know that? DNA has 2 strands. Okay? Now, each one of these letters that are here represents a molecule that's of very critical importance in DNA structure. Okay? I won't go into a whole lot of detail here, but suffice to say, C's are cytosines, G's are guanines, T's are thymines, and A's are adenines. Okay? So, you have these 2 strands, and there's some things I do want to mention very quickly. There are 2 strands of the DNA. One of them is referred to as the coding strand. This figure likes non-template strand, but I will go with coding strand. The other one is referred to as the template strand. Okay? These are the 2 strands of DNA, and one of them is of very critical importance when we're making RNA. Okay, so here's the idea. It's very important that you understand that cytosine, C, in DNA, cytosine, hydrogen bonds with guanine. Always. There's no exceptions to that. Cytosine, hydrogen bonds with guanine. Also, notice thymine, hydrogen bonds with adenine. And it does it 2 times. These 2 interactions right here, these are the fundamental interactions of DNA. That's definitely something you need to know. You need to know it for lab and lecture. The fundamental interactions of DNA are cytosine bonds with guanine and thymine bonds with adenine. Does that make sense? So you notice in this strand, hopefully you can see it. Look at the coding strand, for example. Okay? So the coding strand's sequence, this component of it, goes C, G, C, T, A, T, and so forth, right? Does everybody see that? This is essentially the order of nucleotides. So, for example, the first nucleotide in the coding strand is a cytosine nucleotide. Okay? The second nucleotide in the coding strand is a guanine nucleotide. And you could keep going on and on like this. But do you see that when the coding strand interacts with the template strand, notice C always interacts with G, G always interacts with C, T always interacts with A, and A always interacts with T. Does that make sense? So these are the fundamental interactions in DNA. Now, one thing I do want to mention about this interaction is it is very strong. Okay? In fact, when you find this DNA in the nucleus, generally, unless the genes are activated, it's always like this. These strands are very tightly bound to one another. Does that make sense? In fact, one interesting note is if you actually do, you can take classes like genetics and cell biology here at school, and you'll do an experiment in those where you have to pull apart the strands. Okay? It actually takes 85 to 90 degrees Celsius of water to pull those strands apart. And obviously, if you're in 90 degrees Celsius water, you're dead. Okay, so these guys are together. Okay? It's very difficult to pull them apart. The only way you can do it, really, is to increase the temperature drastically beyond what life supports. So that means that under normal physiological conditions, it's always in this tightly bound form. Does that make sense? Okay. Now, I have this RNA transcript here. We're not going to worry about that right now. The main thing I do want to make sure that you understand is these guys are very closely interacting. And it's very difficult to pull them apart. However, like I mentioned a few minutes ago before I started this tape, is that enzymes are used by cells to get around slow reactions. Okay? In fact, one way to think about an enzyme is it's a molecular bully. Okay? What does a bully do? A bully forces people to do things they don't want to do, right? So by default, the DNA strands don't like to come apart. So what enzymes can do, and there's one in particular I'll get to in a minute, this enzyme literally forces the strands to come apart. Okay? It's going to literally split the strands apart. Now, if you go watch the video that I have on this integrity, what you would see is essentially the following. Okay? Let's say I have these bases. I'm going to get this, erase this. Okay? Let me essentially give you a copy of this. So I have C, G, CTAT, CTAT, AGC, and that's all we do. And then, of course, you have the complementary bases, the one that's always paired with those. G, C, G, A, T, C, G. Okay? Now, we're going to have to somehow make this RNA transcript. And we're about to get into how you do that. But there's something you have to bear in mind. Okay? There's an enzyme you need to be aware of. This enzyme is called Rna Polymerase. On the exam, on the essays, what I do is I want you to be able to give me a function of this in like one sentence. Okay, so nothing terribly long. We'll get to what it does in a few minutes. But RNA polymerase is what directly makes the RNA strand. Okay, RNA polymerase makes all RNA strands in general. Okay, but here's the problem. These strands are so tightly bound together. Okay. If they're not pulled apart, RNA polymerase can't catalyze the reaction. Does that make sense? It's sort of like, I'm trying to think of a good example. If you, like for example, this is a stupid example, but if I have my hands pressed together really tightly like this, do you think that someone would be able to force this tape thing in the middle of these if I had these together real tight? No. You're not going to be able to take this and kind of force it between my hands if my hands are really tightly bound together like this. So if these strands are in the very tightly bound form, which is how they are at rest, this enzyme, RNA polymerase, has trouble polymerizing an RNA strand. It can't make it. Okay? So here's a fundamental concept. Okay? Before any polymerization of an RNA strand occurs, you have to have the access of an enzyme known as a helicase. Okay? There are lots of helicases around, and there's one part of this complex of enzymes that's going to assemble here. There's a complex of enzymes that's going to assemble right here. One of these enzymes is referred to as a helicase. Okay? Here's the idea behind a helicase. So imagine these bases right here, these nucleotides. Imagine these right here are sort of like if you looked at a zipper on a pants, right? If you look at that, you have those little grooves, right? And imagine DNA helicase is the physical metal zipper, right? And what do you do with a zipper? Well, you unzip it, right? So what happens is, if I move the zipper down like that, what happens? The strands come apart, right? It's just like the zipper on a pants or the zipper on a coat or something, okay? If this enzyme, this is one of the DNA helicases, okay? That's the zipper, the literal zipper, okay? When it moves along the strand of DNA, it unzips your gene. Literally, one of these genes right here, like gene 1 that I drew up on the board. If you expect transcription to occur on it, you've got to unzip the gene. Is that clear to everybody? You have to unzip it. So meaning what has to happen is you literally pull those strands apart. And that is the chief job of this enzyme called helicase. And if you understand that, literally on the test I'll ask you, what's the overall function of the helicase? And your answer should be something along the lines of the helicase unzips the DNA, allowing for binding of RNA polymerase. So RNA polymerase cannot accomplish its reaction unless the helicase unzips the DNA. And literally with unzipping the DNA, these strands are going to come physically apart. In fact, let me do this, and I'll explain it using this figure. This is a really good figure that I have. Actually, hold on a second. Yeah, let me explain a few other things first. Okay, so we'll kind of integrate things in a few minutes. Okay, so for example, let's imagine. So this strand right here on top, what was this? The coding strand, and this is down here, the template strand. Okay, so when these strands come apart, so you have to imagine these strands are now physically separated. Okay. In fact, let me actually show you a picture of what it looks like when they're separated. This might give you some intuition. Notice how these two strands have come apart. Do you see that? Do you see in that picture how the two strands of DNA have come apart? So if you look right here, there's a huge gap between the two DNA strands. Do you see that? There's a humongous gap. And in general, the polymerase enzyme is around this region right there. Okay? So let me point to it so if you're watching this, you can see. The polymerase action is approximately right here where my mouse is. Okay? So here's the idea. You would make the assumption at this point that the strands are unzipped. Okay? And now we're going to look at the function of RNA polymerase. Okay? Strands are unzipped, they're pulled apart. So here's what happens. Okay? So I mentioned that in DNA, the fundamental interactions were cytosine bonds with guanine and thymine binds with adenine. Okay? That certainly works for DNA, but here's the problem with RNA. RNA doesn't have thymine. Okay? I think that's something you would have talked about when you took things like microbiology or I don't know if Intro to Chem talked about that. But here's the idea behind RNA. RNA has 4 bases. Okay, 4 nucleotides. It has adenine, it has guanine and cytosine, but the thymine is replaced with another nucleotide called uracil, U. Okay, so let me show you how this works. Okay, what's essentially going to happen is you have DNA right here and the corresponding RNA base. Actually, let me write that up here. Let me erase some stuff. So here's what's going to happen. Okay? This is how RNA polymerase works, how the bases work. If I have some DNA base, I'm going to show you how it interacts with an RNA base. Okay? So if my base is guanine and DNA, the rules don't change for cytosine and guanine. The corresponding RNA base will be cytosine. If my DNA base is cytosine, the corresponding RNA base is always guanine. So these rules don't change at all. But this is where the problem comes in. The problem is that RNA doesn't have thymine, it has uracil. So if you have thymine as a base in DNA, what do you think the RNA base will be? Adenine, right? So thymine will interact with adenine, but here's the problem. If my DNA base is adenine, now what is the RNA base going to be? Uracil. So when you're talking about the fundamental interactions between DNA and RNA, these are the interactions you need to know. Okay? So having that in mind, let's actually look to see exactly how the RNA transcript is made. Okay? So I've unzipped the genes. A helicase unzipped the genes. Now I'm going to make the RNA strand. So this is the RNA strand that's going to be made, but let's reason through it. So what's going to happen is the DNA template strand is going to be where the polymerase acts. And it's going to add complementary bases. So based on this information right here, the DNA-RNA interactions, what would be the complementary base to this GB? C? Right. So the RNA base will be C. What about for this template strand cytosine? G. What about this G? What about A? U, T? Right? Okay, and you could go on and on with the ones that they have written on here. I didn't draw the whole template strand or coding strand. Okay? So let me do this. Okay, let me actually move this computer over here so it catches my audio. Okay. So in fact, let me erase this. We don't need this anymore. I want to show you something interesting. I don't know if you ever thought about this when you were in biology classes. Look at the coding strand. Let me keep the color the same. The coding strand, so this is coding. Coding strand is the following. C, G, C, T, A, T, A, G, C. Notice what the RNA transcript is. It's almost identical. C, G, C, in this case it's U, A, U, miss something? Oh yeah, A, what did I miss? Oh yeah. Yeah, A, G, C. So this is the coding strand. This is the RNA strand that's made. From just looking at this, what's the only difference between those two strands? Right. Notice how the DNA coding strand has T's, RNA has U's. That's what you want. Those are the only differences that are visible using this method between the DNA coding strand and the RNA transcript. So when I told you earlier that the way the cell, so if I have my cell, right, here's my cell, there's the nucleus, I have my DNA in here, but my ribosomes are out here. Here's the ribosome. How does the cell get around getting the genetic information from the DNA out into the cytosol, out here? There's the cytosol. How does it do it? It makes an RNA strand. So literally, this coding strand, this is actually the physical strand that codes for the protein. We'll see how that works next week. The coding strand is the actual code. So the coding strand codes for, in this case, mRNA. And then the mRNA leaves the nucleus and goes to the ribosome. So does everybody kind of see how the coding strand codes for an RNA? So what you end up getting is the RNA strand. It could be mRNA, it could be tRNA, it could be a number of RNAs. The point is it is an exact copy of the coding strand, except what? Thymines replaced with uracil. Based on this, that's basically all you need to know. Okay? Is basically what I'll do on the, I have a practice problem just like this, and I could ask it. What I'll do is I'll say, well, here's a hypothetical coding strand. That's the question. Here's a hypothetical coding strand. Determine the RNA transcript. So if you know the DNA coding strand is the same as the RNA transcript except for T is replaced with U, that's your answer. So in other words, if I gave you this DNA coding strand on the test, this would be your answer for the RNA transcript. So I do want you to be able to go from the DNA coding strand to the RNA. Does that make sense? Okay. So now here comes the question, why do we care about that RNA transcript? Well, the RNA transcript, number one, is used by the ribosome to make a protein. That's week 2. Okay, don't worry about that now. What I'm concerned about right now is the RNA. Okay? The enzyme that does this. So this is pretty typical nomenclature for an enzymatic reaction. What they'll do is they'll essentially have what's the substrate or the reactants, and they'll draw an arrow towards the products. In this case, the product is the RNA transcript. The enzyme that does this is Rna Polymerase. So here's what you need to bear in mind. Helicase unzips the gene. Is that clear to everybody at this point? Helicase pulls the strands apart. Rna Polymerase makes the RNA strand. Okay? That's the intuition you need to keep. So, if I asked you on the test, which I probably will, what is the function of DNA helicase? What I'll do is I'll give you something like this. I will physically give you the coding strand and the template strand. In fact, hint, I might give you this exact picture. So you might want to star this picture if you can. So I might give you that exact picture. I'll tell you what the coding strand is. And I'll say, what does DNA helicase do? What's its function? And what you should probably do is draw the zipper and explain that DNA helicase unzips the genes by pulling the two strands apart. That's basically a one-sentence answer, and that will suffice. Full credit. Then I'm going to ask you, what's the function of RNA polymerase? And what you would need to do in that case is I'll ask you to determine the RNA transcript. So you would know that the RNA transcript is the same as the coding strand, except what? T is replaced with U, right? And then you have the RNA transcript. You just basically take the coding strand and replace all the T's with U's. And when you give the function of RNA polymerase, in one sentence you would probably say something along the lines of RNA polymerase, once the DNA is unzipped, RNA polymerase takes the DNA coding strand and makes an RNA strand. That's your answer. Okay? But I want you to at least have the understanding of how it works. Okay? So if that makes sense to you, that will suffice for the exam. Now, test yourself. So we have some more of these. Okay? So what I'm going to show you before we do this practice problem is this is a picture, a very good picture that I got that shows you basically how RNA polymerase works. So what you should notice from this picture is, notice how the DNA is currently unzipped. Do you see how the strands have come physically apart? The DNA is unzipped. This green strand right here, this is the RNA. Okay, does that make sense? This is the RNA. What I want you to, and notice, the RNA, the bases are being added at this end. Okay? Right where my mouse is, that's where the RNA bases are being added. The nucleotides are being added at this point. But here's something to notice. Notice that the RNA strand, for about 8 bases, remains interacting with the DNA. Do you see that? Do you see how the RNA remains bound to the DNA for just a little bit? It's about 8 base pairs, and I do want you to know that number. So for a short time, the DNA and RNA will interact. Okay? And this interaction between RNA and DNA, where they kind of stick to each other, is called the Dna Rna Hybrid. Okay? Does everyone understand that? So literally when I drew this sequence right here, let's see, 1, 2, 3, 4, 5, 6, 7, 8. Maybe this part of the RNA is bound to the template strand. Okay? But maybe this one right here is not. Okay? Does that make sense? So for about 8 base pairs, the RNA will remain interacting with the DNA. That's called the Rna-DNA hybrid. So my question in the practice problem is the following. In the Dna Rna hybrid, adenine forms, and you're going to use this picture right here, adenine forms how many hydrogen bonds with what? So first of all, looking at this picture, if the DNA is adenine, what nucleotide does it bond to? Uracil. So A always goes with U. You can see that right here. Here's an AU. Okay? A always bonds with U. Okay? How many hydrogen bonds are formed? 2. Exactly. So T and A always are 2 hydrogen bonds. A and U are always 2 hydrogen bonds. G and C don't change at all. They're always 3. Okay? So anytime the DNA has an A, the RNA base that goes with it is always a uracil, and it's 2 hydrogen bonds. Okay? I know that your book in lecture will elucidate that to you. They'll expect you to know this fundamental interaction right here. They will expect you to know that. Okay? So in the DNA RNA hybrid, adenine forms 2 hydrogen bonds with uracil. Second one, okay? So this will be good practice. In fact, this right here, this question, you should star this. I'll tell you this is on your test. This question's on your test, okay? So what I'm going to do is since I don't have the marker, I'm going to copy this down. So it is A, C, G, T, T, A, C, G, A, G, C. Let's just carry it out that much. It's pretty long. So what do they tell us? Well, the following sequence of nucleotides is the coding strand of some part of the DNA molecule. Well, let's go back to one of the other slides. This top one is the coding strand. So what did I say was the only difference between the coding strand and the RNA strand? What is it? Right, T is replaced with U. So if this is in fact the coding strand, how do I get the RNA transcript? Exactly. That's exactly correct. So anywhere I see a T, I just put a U. And everything else is exactly the same. And there's 3 others, I think, that I didn't have room for. This would be your answer. So you would say this right here, this is the RNA transcript. This is your answer. Does that make sense? So what I'll do on the test is I'll give you the coding strand. I'll probably change the sequence a little bit, or I could give you the one on this slide. I could give you that, and I could just say determine the RNA transcript. And literally what you do with the coding strand is you take all the T's and replace them with U's, and that's your answer. Does that make sense? This RNA transcript, we're going to use this next week. We're not going to use it this week, but you need to have it sort of filed away that we made an RNA transcript, and then we're actually going to use this, assuming this was mRNA. We're going to use this to make a protein, and that process is called translation. It's called translation, and it's carried out by an enzyme known as the ribosome. Okay? And that process is translation. Okay? So hopefully that makes a little bit of sense. And now let's actually look at the picture. Well, let's look at a few pictures that kind of tie everything together. Let's go through this stuff one more time. Okay? So I'm looking at this picture right here on the top. These are the DNA strands. Okay? This brown thing in here that kind of looks like a potato, this right here, that's the helicase. Do you see how it just broke the 2 strands apart? Does that make sense? So the helicase in this case is moving right to left. Okay, it's moving right to left, and in the process, it's basically ripping those strands apart. Do you see that? That's the job of the helicase. And specifically what it's doing is it's breaking the hydrogen bonds. Because remember, when I looked at these DNA fundamental interactions, cytosine hydrogen bonds 3 times with guanine, thymine hydrogen bonds 2 times with adenine. What helicase is doing is it's breaking those apart. Does that make sense to everybody? So helicase rips the 2 strands apart. Okay? And helicase is one of the 3 enzymes you need to know. One of them we haven't gotten to yet. Then RNA polymerase. Okay? What you need to know from this slide, essentially, is the following. Once you have the strands separated using the helicase activity of the complex, you take the template strand, right here, this is the template strand, and you add complementary RNA bases. So everywhere I see a G, the RNA base is a C that goes with it. Anywhere there's a C, the RNA complementary base is G. Okay? And so you make this RNA strand right here, and what you could also say in the answer is the RNA transcript is an exact copy of the DNA coding strand, except thymines replaced with uracil. Okay? And what you need to do when you're doing this is, print out those practice problems and go through those. Because likely what's going to happen is, actually not likely, exactly what I'm going to happen is, I'm not going to change the questions at all. I'm literally going to take those essays and copy and paste them onto the exam. So you're going to learn all this using those essays, okay? and you're literally just I'm going to copy and paste those right into the test. Does that make sense? So that's basically RNA polymerase. And at this point, what we've seen is we've seen the synthesis of the mRNA transcript, or really any RNA transcript in general. Okay. Is it 447? 447. Okay. So while you're packing up, I want to show you just one more thing, and then we'll call it, because we really don't need anything else for the most part. Okay. One of the things that's really interesting about, and let me see if I still have access to it. It might have logged me out. No, in fact, what I'll do, the rest of this that we need, I'll just make a video for it, and you're welcome to watch it. Okay. Like I said, anything that you need is going to be taught in some way. I'll shoot whenever I post a video or find one that's useful online. I will definitely send an email out through Blackboard to your patron emails. Okay, does that make sense? So on some way it will be taught. You'll see it in lecture, and make sure to go to Blackboard and print out those essays. That's going to be basically your test. Okay, those are the things I think are important, and hopefully dealing with the models and stuff and this PowerPoint right here, hopefully you kind of got an intuitive sense of what I'm going to ask. Okay, so that's basically all we have time for. Like I said, I'm going to be